All right, good afternoon, good morning to everyone. Thank you so much for joining us today for another HDSA research webinar. And if you are joining us for HDSA's virtual convention, where this will also be airing, welcome to our research day. Joining us today are Dr. Enchi Liu and Dr. Sanjay Keswani from Annexon Biosciences, who I will introduce in just a moment. But first, I want to remind you how this works, how to ask questions in this webinar. So we will be doing Q&A after the presentation with Dr. Liu and Dr. Keswani, but you can send a question anytime during the presentation by using the chat function in your toolbar. You can find that toolbar either on the top or bottom of your screen. And you can click on that chat function, type in your question, hit send, and only HDSA and the panelists are going to be able to see your question. And we will do as many questions as we can at the end. And if you are joining us for convention, or if you're just joining us on your Tuesday and you're looking forward to convention, I wanted to mention all of the other great presentations that we have lined up for our research forum on Saturday, June 6th. These times are in Eastern Time. At 11 a.m., Dr. George Yorling from HDSA is going to welcome everyone. We will have Wave Life Sciences speaking about their research at 11:15. At 12:15, Roche Genentech is going to be talking about their clinical program. We'll have an HD Buzz Year in Review at 1 p.m. from Dr. Jeff Carroll and Dr. Ed Wild. And at 2:30 p.m., we'll have a clinical trials showcase with some other biotech and research companies that are working on Huntington's disease. All of these sessions are free. Everything at convention is free. Registration is required for a lot of these sessions, so please go to hdsa.org/convention to see what else is going on with research, not just in the research forum, but the whole week. So I also want to remind you that this presentation is being recorded for convention and just for viewing later. So you can go to our research webinars page at hdsa.org/researchwebinar, or you can go to HDSA's YouTube channel, which you can find at the top of our homepage. And usually we try and get these webinars and presentations up within a couple of days as we can. And so now I would like to introduce everybody. Also joining us is Dr. George Yorling, Chief Scientific Officer and Chief Mission Officer at HDSA. If you want to say hi, George.
Hi, everyone. Thank you for joining us. Excited to hear from this wonderful presentation from Annexon. Hope everyone's well and staying safe.
Thanks, George. And now I would like to introduce Dr. Sanjay Keswani and Dr. Enchi Liu. So Dr. Sanjay Keswani joined Annexon Biosciences as Chief Medical Officer in June of 2019. He's an accomplished physician scientist with broad research and development expertise in multiple therapeutic areas, and is a former neurology faculty member at Johns Hopkins and an elected fellow of the Royal College of Physicians in the UK. Dr. Keswani has held senior executive positions in the pharmaceutical industry, including Roche, where he was Senior Vice President and Global Head of Neuroscience, Ophthalmology, and Rare Diseases Research and Early Development. During his tenure at Roche, his group successfully advanced 5 molecules to phase 3 development. And prior to Roche, he was Vice President, Exploratory and Clinical Translational Research at Bristol Myers Squibb, where he was responsible for multiple therapeutic areas, including immunology, neuroscience, rare diseases, fibrosis, and virology. He also held R&D leadership roles at Eli Lilly and Company and Amgen, where he prosecuted the development of the now approved CGRP antibodies for migraine prevention, Emgality and Aimovig. Dr. Keswani completed his residency in neurology and fellowships in neuroimmunology and neurophysiology at Johns Hopkins, where he cared for patients with diverse neurological diseases and ran an R01-funded neuroimmunology research lab. He graduated in medicine at St. Bartholomew's Hospital and holds a first-class honors BSc degree from St. Mary's Hospital, London, in pathology and basic medical sciences and immunology. Dr. Enchi Liu has extensive experience in drug development, and in addition to working on Huntington's disease, she's worked on therapies targeting mood, pain, sleep, Alzheimer's, ALS, Parkinson's, and several rare diseases. Her expertise extends to implementation of biomarkers for translational research and to support development candidate registration. Prior to Annexon, she served as VP Head of Clinical Development and Translational Biomarkers at Escape Bio, Program Team Leader for a Rare Disease Program at Prothena Biosciences, Alzheimer's Disease Biomarker Team Leader at Janssen Alzheimer Immunotherapy and Janssen Research and Development, and Senior Director responsible for leading the Alzheimer's clinical development and operation teams at Oran Pharmaceuticals. Dr. Liu received her PhD in neurobiology and behavior from Cornell University, where she conducted research on cellular and molecular biology of neurotransmitter receptors. So we are very pleased to welcome these two very accomplished speakers today, and now I'm going to hand it over to them.
Thank you. Let's see. Share screen. Just checking that, Leora, you can see my screen, correct?
Yep, you can see.
Okay.
Well, thank you, Angie.
Great.
So good morning and good afternoon, everyone. Really like to thank the Huntington's Disease Society of America for giving us the opportunity to talk about our complement inhibitor in the context of treating neurodegenerative diseases such as Huntington's disease. Just move on to the next slide.
I may have to get out of, hold on.
Maybe while Angie's pulling up the slides, I could speak a bit about the introduction in terms of how the company was founded. So essentially, it was founded by Ben Barris's seminal work in the complement pathway, which is typically viewed as an inflammatory pathway. But what he showed in his seminal findings, he incidentally was the chair of neurobiology at Stanford, was that a protein in the classical complement pathway called C1q was really important from a developmental point of view, and actually it's on this slide. And C1q was also a major player in the context of causing synaptic loss in a variety of neurodegenerative diseases. And essentially that spawned Annexon, and we're targeting C1q, and I'll explain a bit about why we think that may be helpful in patients with neurodegenerative diseases, including Huntington's. And we can move to the next slide. So having an anti-C1q antibody allows us to target a wide variety of diseases. And these include antibody-mediated autoimmune diseases, which I won't speak about much for the rest of the talk, because I'll focus on the second pathway. Oh, if we just go back, Angie. Yeah.
Yep.
So antibody-mediated diseases include hemolytic anemias and lupus. So those are diseases that we're targeting. But what we'll focus the rest of the talk on is complement-mediated neurodegeneration. And this is this unique hypothesis, again, inspired by Ben Barris. Yeah, we can move on to the next slide as I'll explain more about that. And so the rest of the talk is really divided into 3 bits. If you just go back one slide, Angie, helps. A background on complement-mediated neurodegeneration. And then I'll speak a bit about the efficacy of anti-C1q in animal models of neurodegeneration. And then lastly, focus specifically on Huntington's disease and our clinical program. Let's move on to the next slide. So I'll be speaking about synapses quite a lot. So I thought it'd be good to have just an introductory slide on this. And the details aren't so important here, except for really 2 key points. Firstly, that synapses are the major points of intersectional communication between neurons. Essentially, it's how neurons communicate with each other, and also how neurons communicate with parts of the body, such as muscles. So they're really important for preservation of cognitive function, as well as motor function as well. This really introduces Ben Barris's work on the role of C1q. There are three figures here, and I'll start with the left figure in the developing brain. So his work essentially showed that in normal development of the brain, we need to get rid of synapses that are not relevant to fine-tune our neural circuits. So C1q plays a role in tagging those synapses for elimination. And the cells that cause the elimination are those yellow cells at the bottom, the microglial cells, which essentially eat the synapse once it's been tagged by C1q. So that's all normal because that's the way to get rid of excess synapses. And again, promote a finely tuned neuronal circuit. Now, once we have matured and are adults, then the C1q system is pretty silent. And so that's the figure in the middle. So there's not much action going on. C1q is not labeling synapses, and synapses are not being lost. However, in neurodegenerative diseases, and that's that figure on the right, this C1q pathway becomes aberrantly triggered, such that synapses that are functional now become triggered with respect to elimination by these yellow microglial cells. And this is essentially what we're trying to do at Annexon, is to stop this aberrant C1q activity and hence prevent synaptic elimination and therefore prevent neurological disability in patients. And this is some mouse work. This is a mouse model where C1q was looked at by Ben Barris and Beth Stevens. And essentially, if one looks on the graph on the right, C1q knockout animals, so these are mice which don't have C1q because of a genetic deletion, have more synapses, nearly twice as many synapses as wild-type mice or WT mice, which have normal C1q levels. Interestingly, these knockout animals also seem to be preserved from age-related cognitive deficits. So that was interesting in its own right. Go on to the next slide, Angie. Now, what's kind of puzzled individuals who've researched neuroscience over the years is why aging is a big risk factor for neurodegeneration. Because we know that for many diseases, Alzheimer's disease, Huntington's, that disease manifests later in life. Parkinson's disease is another example where disease happens later. So why is that? And the other interesting question for neuroscientists is why certain regions of the brain seem more vulnerable to neurodegeneration. Again, a hypothesis to explain that is C1q accumulation. And this is depicted on the graph on the right. So in aged animals, in this case, these were aged mice, C1q accumulates enormously. So there's a 300-fold elevation in these aged brains. And this was confirmed in human brains as well. So this was interesting in terms of maybe this being a risk factor for neurodegeneration as one gets older. The other finding that was notable was that this C1q accumulation was regional and seemed to be in regions of the brain that were particularly vulnerable to neurodegeneration. So for example, the hippocampus, which is important in learning and memory, was a major region that was affected. In the substantia nigra was another region, which is an important region for motor control. So this also may be an explanation for why certain brain regions are more vulnerable to neurodegeneration than other brain regions. Let's move to the next slide, Angie.
Yeah, there it goes.
Great. And so now just move on to talk about the efficacy of anti-C1q in a variety of animal models of neurodegeneration. So at Annexon, we're essentially targeting C1q. What we're trying to do here is prevent C1q binding to the surface of the synapse, and therefore prevent activation of this inflammatory cascade, the complement pathway, and therefore prevent elimination of these synapses and prevention of neurological disability. We move to the next slide. This is just a really high-level introduction of the complement pathway, which is a rather complicated pathway. There's just some key messages on this slide. So C1q is the initiating molecule of the classical complement pathway. And so by blocking C1q, one blocks all downstream components of this pathway. And components of this pathway include immune cell recruitment. So that's infiltration by cells such as microglia, which I mentioned are the cells that kind of eat the synapses. Immune cell attack, and that also relates to microglial phagocytosis or eating, as well as membrane damage. And these are damage directly to the neuronal cell membranes via the so-called membrane attack complex. Now, what we thought was really exciting, this is work via Ben Barres, but many other investigators across the world, that C1q inhibition protects against synaptic loss from a wide variety of triggers. This was shown in models of Huntington's disease, Alzheimer's, spinal muscular atrophy, and eye disease, glaucoma, where retinal ganglion cells are lost, and that results in visual field loss. Geographic atrophy, frontotemporal dementia, and traumatic brain injury and stroke. And I mention all these models just to illustrate how widespread this protective effect is. And so what we believe is that regardless of the upstream cause or trigger, at the end of the day, the reason patients become disabled with respect to motor dysfunction and cognition is because they're losing synapses. If one can prevent synaptic elimination from C1q, then one may be able to prevent neurological disability across a wide variety of diseases. And so this is our core belief, our core hypothesis, that clearly we would like to test in the clinic. So this is just some examples of this efficacy data. So here are two models, Huntington's disease on the left, and I'll speak a bit more about this actually, and spinal muscular atrophy on the right. Now, just for introduction, SMA or spinal muscular atrophy is a severe disease affecting the neuromuscular system, and it affects children particularly. And so in both these disease models, we were able to protect against synapse loss via blocking C1q. And specifically, the work on the left was via Beth Stevens and Dan Wilton at Harvard. And the work on the right was recently published in Cell Reports by George Mentz's group at Columbia University. And this group showed, in addition to protecting against synaptic loss, that motor weakness was also ameliorated in animals given anti-C1q. Now, I'd like to focus on our Huntington's disease program with 005. So again, this audience doesn't need much of introduction to Huntington's disease. What I'd like to mention here on this slide is that neurofilament light chain is an important biomarker for us, and it's known that Huntington's disease patients have high baseline NFL levels, and the reason that's important is because we're intending to use NFL as a marker of neurodegeneration, and to see if our drug is actually impacting neurodegeneration in real time in patients. And also that the classical pathway, and specifically C1q, we believe is highly relevant to pathophysiology, and this is via samples that we'd collected from CSF from patients. Let's move to the next slide, actually. Yeah, and this is a slide that summarizes our scientific rationale. We've mentioned the importance of synaptic loss and how it drives disability. we have data showing that C1q is associated with this synapse loss in both patient tissue as well as mouse models of HD. And I'll show you some of that actually on the next slide. I'll also show you that NFL is elevated in HD patients and does correlate with disease progression. Much of that work was done actually by Ed Wild's group at University College London. And also, by giving an anti-C1q antibody, we're able to reduce synapse loss and also reduce neurofilament light chain, which is this biomarker of neurodegeneration in mouse models of Huntington's disease. And then just lastly, having natural history cohorts to provide information on the disease is just so important from drug development. And this is really a reflection of how important patients are and their families with respect to contributing data that will really help drug development in this area. This highlights the synapse loss that's evident in both Huntington's disease patients, and this is from post-mortem brain tissue that's on the left, and also reflected in a mouse model of Huntington's disease on the right. So essentially, these graphs are both showing that as disease progresses, synapses are being lost. And these synapses are specific ones. They're so-called corticostriatal synapses. And these synapses are really important with respect to control of motor movements, which we know are impaired in patients with Huntington's disease. Now, coincident with this synaptic loss is evidence for increased complement activation in the brain tissue from Huntington's disease patients. And this was work, again, via Dan Wilton and Beth Stevens at Harvard. Those are the graphs on the left of the slide showing that C3 and a cleavage product of C3, which is IC3B, is present on synapses in brains from Huntington's disease patients at a much higher level than you'd expect when you measure against control individuals. Now, via a collaboration with Ed Wild at Queen Square, and that's reflected in the middle and the right panels, levels of C4A, which is another component of the classical complement pathway, are elevated in the CSF or cerebrospinal fluid from patients with Huntington's disease. And that seems to correlate as the disease advances. So as patients become more symptomatic, then C4A levels in CSF increase. And on the right, we were interested to note that C4A correlated with multiple clinical endpoints, both measuring cognition as well as motor function. This slide's really introduction of neurofilament light chain, or NFL. So NFL is a neuronal cytoplasmic protein. So it's actually present on the inside of neurons, and their projections called axons. There's a lot of excitement in the field of neuroscience across many diseases, because now we can measure NFL quite sensitively at very low levels. We couldn't detect it earlier with the older assays. And that's resulted in a lot of longitudinal studies showing that NFL levels correlate with disease severity and also have important prognostic outcomes. And this is in many different diseases, for example, multiple sclerosis or spinal muscular atrophy, etc. But also the case for Huntington's disease. And again, a lot of this was via Ed Wild's data at Queen Square. Essentially, and actually I think this is on the next slide. Oh, actually, I think we skipped a slide here, Enchi, if you go back.
Yeah.
Yeah, great. Okay, so and I'll start actually on the right with the human data. Again, this is via Dr. Byrne as well and Dr. Wild at University of London. What they showed was that neurofilament light chain increased as patients became asymptomatic. They were also present in pre-HD patients before they were symptomatic, and then after those symptomatic as well, where they increased further. And the levels of NFL in the CSF also correlated with total motor score, i.e. the higher the NFL level, the worse the motor deficits. This is illustrated in the graph on the right. And of note, with respect to a mouse model of HD, the R62 model, one can recapitulate this increase of NFL in the CSF from these animals. And that's important because one can then use this mouse model to detect if you're reducing NFL. Move on to the next slide. And indeed, this is what we did. And so with the application of 005 and anti-C1q antibody in this mouse model of Huntington's disease, we're able to show a decrease in complement deposition on synapses. And this was associated with a decrease in neurofilament light chain in the CSF. That was actually after 2 months of treatment. This was associated with some preservation of synapse number as well, so some protection against synapse loss. So in summary, we're pretty encouraged by the data that we have, both from the original Ben Barris work and then other investigators confirming the predictive role of anti-C1q in a variety of animal models of neurodegeneration, including Huntington's disease. We're therefore initiating a phase 2A clinical trial of Huntington's disease later this year. And we've actually activated the IND for this study with the FDA. And this is a US based trial. It's a relatively small trial, so about 20 to 24 patients, and it's open label, i.e. everyone would get the drug. It's a relatively short duration study, so 3 months treatment duration, 3 months follow-up, and it's a biomarker study. It's really looking at the change of key biomarkers after this relatively short duration of treatment. Specifically, these are biomarkers that measure CNS target engagement. So it's important for us to understand when we give this antibody intravenously, how much of it's actually getting into the brain and actually hitting the target, which in this case is C1q. And we can measure that target engagement by taking CSF from patients, this is via lumbar punctures, to understand if we're impacting target actually within the brain. We're also looking for reduction of this important neuron-specific biomarker, which reflects neurodegeneration, called NFL. What we'd like to understand is whether our drug candidate impacts neurofilament light chain. Specifically, does it reduce NFL from baseline in these patients? Clearly, drug development takes an enormous effort with many partners. We're fortunate to have established partnerships with key academic consortia and patient advocacy groups, such as the Huntington Study Group, and clearly very excited to partner with HDSA as well as Enroll HD as well. This data we think will be very informative with respect to helping us understand whether it would be worth pursuing a formal efficacy and registrational program in Huntington's disease. So this is an important clinical study for us. And I think with that, ends my presentation, and Enchi and I would be happy to address any questions that you may have.
Great, thank you so much, Dr. Keswani and Dr. Liu too, for sharing this data with the community. I'll just give some more folks time to type in their questions, but I also wanted to mention that at HDSA we were particularly excited to hear from you today, because HDSA's Human Biology Project actually supported Dan Wilton working with Beth Stevens on some of the early work that led to this. So we've got some questions coming in. One person wants to know a little bit more about your specific endpoints when it comes to the biomarkers you're looking at.
Sure, I can take that. So I think just firstly, in terms of this phase 2A study, we want to make sure that the drug is safe. So safety and tolerability is important in this study. We'd also be looking for biomarkers of neurodegeneration. So I mentioned neurofilament light chain. So we'd be measuring NFL in spinal fluid from patients both at baseline and after treatment. And also another biomarker is measuring so-called free C1q levels, again in CSF from patients, ensuring that the drug that we're giving intravenously actually gets into the brain and inhibits target. So one way we can understand that is by measuring those biomarkers of target engagement in the CSF. We're also looking in an exploratory fashion at quantitative EEG, which is potentially a marker of synaptic function as it relates to the cortex. And so we're looking at quantitative EEG both at baseline as well as after treatment. So those are, I think, the major biomarkers. Enshi, I don't know if there's something more that you'd like to add there.
No, that's perfect. Thank you.
Yeah, thanks for expanding upon that. Another person asks, is wondering at what stage of HD would you anticipate this drug being delivered?
Yeah, it's always a tricky question, and it's somewhat hypothetical because the best place to target a therapeutic, I think, will be borne out by clinical trial data. We're encouraged with this particular hypothesis because we feel that synaptic loss is a driver of ongoing disability in patients, and hence we could intervene, relatively at a later time point than potentially other therapies, which are targeting, the upstream cause. Clearly, the earlier one treats, the better, and I think that would be applicable to our treatment. And so, for example, in the phase 2A trial, we're enrolling individuals who have, early manifest HD, particularly, I think. But Enshi can speak a bit more about that.
No, that's right. I think we would like to, in the end, be able to treat all stages of the disease. But in this initial study, we're looking to enroll early manifest Huntington's patients because it will inform our future potential pivotal trials.
We have a couple more questions around that. I know that there are sometimes different definitions of what early manifest can look like. So we've definitely got a couple questions around exclusion, inclusion criteria in terms of what early manifest means there. And then just if you could talk a little bit more about the trial. We've got a bunch of people asking about details around that, if you're able to share anything like that, such as how can people get involved and whether you have sites planned, how to, where it might be taking place.
Leora, I can take that, and maybe Sanjay could add to that. The details of the study protocol, of course, can be shared with anyone interested in participating with a participating study physician. But briefly, in terms of early manifest, there are some criteria, but I think it would be best if you spoke with a study physician. In terms of the study sites, that information can be available through HDSA, or when we post on clinicaltrials.gov, those details will be available. But I can say that right now we're focusing on US sites up to about 12 centers around the country.
Thanks. Yeah, so there are some people asking specific questions about the trial and the frequency of dosage and benefits, et cetera. So it sounds like that information will be forthcoming as things progress on your end.
Yes.
And someone wants to know how you might be working with nonprofits and patient advocates, whether that kind of input is going to go into the trial design to make sure it fits needs of the community.
Yeah, unfortunately we're not meeting in person this year, but last year Annexon Biosciences attended the Huntington study group meeting back in November. We had a patient advocacy forum where we talked about the synopsis and design. We also spoke to many of the research centers that are part of the Huntington study group. So we've incorporated already a lot of input both from researchers and patients into the synopsis.
That's great to hear.
Yeah, I mean, just
I've got
maybe just
some,
to add to that. So
go ahead.
we clearly would like to get more engaged with patient advocacy groups, and we're a small company, and clearly, you know, there's so much important work going on, and particularly as, you know, hopefully when we move into potentially registrational work, having that input in terms of the appropriate design and feasibility is going to be key. I think for this relatively small biomarker study, we did talk to Huntington study group, and they helped us a bit in terms of the design of the study. It's obviously an important study in terms of understanding whether our drug would have value for Huntington's disease patients, and would be a gate for you know, future much larger studies.
Thank you. Got a couple of more scientific questions here. One person is asking, so you talked a little bit about pruning synapses and the complement system as part of the immune system, and someone's wondering if this is related to autophagy, the idea of sort of self-eating inside of cells.
Yeah, well, wow, that's a pretty specific question, and hopefully I'll do a reasonable answer, and Enchi, you can help out. I don't think so. At least I think they're independent concepts. So here, rather than the cell kind of digesting itself almost, here a microglial cell is essentially eating a synapse that is tagged by C1q. And so that is, you know, fairly typical phagocytosis and engulfment, and relates to, you know, an important role of microglial cells in terms of normally just clearing up debris, but in this particular case, actually eliminating synapses. Again, from a developmental point of view, that's perfectly normal, as we want to preserve the best synapses for truly well-functioning neural circuits. But as mentioned, neurodegenerative diseases, neuronal stress results in these synapses becoming somewhat sticky to C1q, and C1q detects these synapses, and then this aberrantly triggers this developmental pathway of synaptic elimination.
Yeah, Sanjay, I'd just like to add that it might be akin to C1q tagging a synapse and recruiting these microglia cells that their normal function is to clear debris, but the synapse is now being tagged as debris. So it's a recruitment of a different cell type to, you know, eliminate the synapses. Maybe that helps too. So.
Yeah, there seems to be a little bit of a distinction between those two symptoms, those two systems. We have some questions about neurofilament light chain. One question is whether the level of NFL in CSF can be used to measure progression of HD in someone who's pre-symptomatic. And then with ANX005, was that decrease in NFL related to the size of the CAG repeat at all?
Yeah, both good questions. So with respect to the first, yeah, there's I think a lot of excitement with respect to utilizing NFL as a longitudinal tool to understand progression, and hopefully the impact of drugs would have on that progression. And that's how we're viewing it. So in this phase 2A study, you know, we're hoping to replicate what we saw in that mouse model of Huntington's disease, where we're able to have a reduction in NFL, suggesting to us that, yes, we are actually impacting neurodegeneration, and that should result, therefore, in less disability accrual. The question, I think, related also to specifically pre-symptomatic individuals. So I think what you know, Ed Wild's group showed nicely was that individuals who are pre-symptomatic still have elevated levels, and so potentially even pre-symptomatic patients, you know, that might be useful in terms of NFL measures, and certainly that's something we would think about down the road for our drug candidate. The last question I'm just trying to remember now, related to CAG repeats, right? So yeah,
Yes.
this was
Was there a difference in lowering?
so this was done actually in a pretty aggressive transgenic mouse model, the R62 model of Huntington's disease, and so we didn't specifically test, a correlation between CAG repeat and our propensity to reduce. We would expect there wouldn't be much of a correlation though, because we're actually relatively agnostic with respect to the upstream trigger. As I mentioned before, there is efficacy across a wide variety of neurodegenerative disease, even outside Huntington's disease. So at least our hypothesis that regardless of the upstream trigger, and therefore regardless of the size of the CAG repeat specifically, we should be preventing synaptic loss. Again, if this hypothesis is borne out in the clinic.
Thank you. Another sort of related question is why you decided to target C1q instead of C3 or C4 that it seems was shown to correlate with HD progression in particular.
Yeah, those downstream classical component pathways are reflective actually of original C1q activation. So by targeting C1q, we're able to actually stop the train from moving, as it were. I guess if you intervene a bit later on, the train is already moving. And so that's, I think, one component of the answer. The other is that by blocking C1q, one's able to block many upstream and downstream classical component pathways. So I think there was a slide in our presentation with respect to infiltration by immune cells. Some of that is directly C1q mediated, some of that may be C4 mediated, some of that C3 mediated. The point is that by blocking C1q, one would be able to block all these different components.
Thank you. We've got lots of great here. Someone is wondering a little bit more about the agent itself, whether this is a form of gene silencing or if it's something else.
This is actually an intravenous anti-C1q antibody. It's a conventional antibody, and it's given intravenously.
Thank you. Someone is wondering, considering that this drug will hopefully eventually target patients at all stages of the disease, would you expect it to prevent neurodegeneration or potentially reverse degeneration that has already happened?
Yeah, that's a great question. I think that our current thinking, and this is what we'd be testing in our trials probably down the road with respect to efficacy, is prevention of accrual of disability. And by treating early enough, that would be very meaningful to patients. I think it's an open question with respect to kind of reversal of deficit. I think a lot of it relates to the endogenous ability of the brain to repair itself. And again, it's very theoretical, but one might believe that if one is shutting down the degenerative cascade, then it allows more potential for one's endogenous reparative process to occur. But that would be a much higher bar for this therapeutic. And at the moment we're thinking prevention is probably where we'll initially kind of test our therapeutic.
That makes sense. And someone's wondering if there are other clinical studies going on right now of ANX005 for other diseases, dementia, Alzheimer's,
yeah,
ALS.
So we have another phase 2 study starting this year in ALS. That's with 005, this intravenous anti-C1q antibody. And also we have other studies, other diseases that we're targeting with this drug candidate, specifically Guillain-Barré syndrome, which is an antibody-mediated neurological disease. We're starting a potentially pivotal or registrational program in that indication later this year. And then we're also targeting some autoimmune diseases, such as warm autoimmune hemolytic anemia, which is a rare hemolytic anemia caused by autoantibodies. And so, yeah, there's quite a few indications that we're currently targeting. Down the road, clearly, if we see efficacy and signals in these initial neurological diseases, i.e., Huntington's disease and ALS, then clearly other diseases would be in scope for us, such as Alzheimer's disease, which is where Ben Barris did a lot of his seminal work, and frontotemporal dementias, progressive MS, etc. So, we have lofty aspirations, but clearly we just want to make sure that we're sensible in terms of how we stage our drug development process. And so these studies in Huntington's disease will be very important for Huntington's disease itself, but potentially other neurodegenerative diseases down the road.
Right, and you mentioned that that you're targeting different autoimmune diseases, and that the complement system is really a part of the immune system. So does that mean that targeting this pathway could cause immune system side effects for someone with Huntington's disease or another neurodegenerative disorder?
Yeah, that's a great question, and indeed, I think when when interested patients and their families talk to study investigators, that I think would be an important topic, and that's detailed clearly in the investigator brochure and the protocol. So one of the consequences of inhibiting complement may be an increased tendency to infections, specifically encapsulated pathogens. So there are a certain number of bacteria, there may be an increased risk. And so particularly in the context of chronic administration, so when we're giving the drug repeatedly, we are asking that individuals either be vaccinated or have documentation of vaccination against these bacteria. And that's really as a precaution. I mean, we haven't ourselves seen any infections due to our drug so far in our clinical programs. But that's something that clearly we're going to be cognizant with respect to monitoring for in all our upcoming studies, particularly as our studies get longer and longer. So again, we haven't seen it yet, but that's something that we're going to be clearly monitoring for. And we think vaccination will be important in prevention. There are other complement antagonist inhibitors, that actually already approved. And so they give some, I guess, reassurance that these infections can be managed effectively and prevented. And so we're utilizing information from these other complement antagonist drugs that have gone before us.
That is sort of related to another question that has come in, which is it's also pretty specific, whether there might be differences in response that could be linked to genetic variation in complement.
I don't know, Enchi, if you have any specific thoughts. As far as we know, there aren't any frequent kind of polymorphisms that we would specifically focus or enrich our studies in Huntington's disease. And because we're interfering downstream in what we think is a common effect or pathway for a variety of upstream causes, we're pretty agnostic in terms of the patients that we bring into the study. We mentioned CAG repeats, for example. I mean, I think that would largely relate to the risk of progression in a clinical study, and so that would be more relevant rather than the lack of responsiveness, as it were. I don't know, Enchi, if we've thought about complement polymorphisms or genetics in our...
No, and right now we're not stratifying or excluding anyone with different polymorphisms. Like you said, we're targeting something downstream of the etiology of the disease, and maybe within a larger trial, we can look at the data and find something interesting. But right now, we're not excluding people based on polymorphisms in C1q.
Okay, thanks. We've got some questions coming in about juvenile Huntington's disease. People are interested in whether this would include treatment for people with JHD. And in general, let's see if I can sum up a couple of questions. So since the complement, well, if pruning synapses is important when people are young, really, what would that mean for this approach with respect to juvenile Huntington's disease?
Yeah, that's a really important question. Yeah, when we're thinking about brain development, we're thinking about really young children and babies. But to be candid, that's not something I've specifically thought about. Maybe Enshi's thought about. I mean, clearly down the road, we would really like to be as inclusive as possible in terms of HD patients, including juvenile Huntington's patients. That's a great question in terms of normal brain development. But I think that would affect, that would be a concern for very young patients. Actually, I don't know if you have.
Yeah, no, I mean, this is a really good question, and I think Annexon Biosciences will be incorporated into our thinking for our clinical program. But specifically, this initial trial will be in people 18 years and older. But we'll do our homework and think about whether there's an age range below 18 that we can, take this approach.
Thank you. How about one more scientific question here? How are you addressing the blood-brain barrier in this treatment? Maybe a little bit more.
Go ahead.
Sorry, go ahead. No, I was just going to say maybe a little bit more about how the drug gets into the brain.
Yeah. So there are two components to that answer. The first is that we are utilizing relatively high doses of the intravenous antibody to make sure that enough gets across the blood-brain barrier. We do know that there are 1,000-fold less C1q levels in the brain than in the blood. And we've actually measured CSF levels of C1q as well as in blood in preclinical species, but also in patients with GBS. That was the Guillain-Barré syndrome population that we did phase 1b studies in. And so we were able to completely suppress target in CSF from patients with GBS, but also in preclinical species as well who have an intact blood-brain barrier. So just in response to the question, there's nothing specific about our antibody that makes it cross the blood-brain barrier better, but we feel by dosing appropriately, and also because the target concentrations are so much less than in blood, that we should be able to engage target at the doses we're giving in our clinical studies. But that was obviously a very important consideration with respect to how we choose our doses in the clinic. Right.
I have another clinical question here. Maybe there's a couple of questions here that I can sum up. So basically, beyond the decrease in synapse loss, are there any indications on what kind of impact on disease symptoms that you might expect? So, for example, have there been any promising behavioral changes in any kinds of animal models, or what kinds of changes would you expect if you were to treat someone with this drug, hypothetically?
Yeah, I mean, hypothetically, because synapses are prevalent, frankly, everywhere with respect to neural circuits that control cognition, mood and affect, motor control, essentially how we all function from the nervous system. We feel that by preventing synapse loss, we would globally prevent disability in all these different domains. We know that early on, the corticostriatal synapse circuit, these are, I guess for the more technically minded, they're excitatory glutamatergic cortical inputs into the dorsal striatum. Those get affected pretty early on in HD, and so much of the work that I talked about with a post-mortem brain tissue, as well as the animal models, actually focused on those circuits, and it just so happens those circuits are the ones that get preferentially drawn to this C1q activator. So I think one could argue that we would have fairly global effects with respect to disability in HD. In terms of what we did preclinically, so we looked at long-term potentiation models. This is, again, via Beth Stevens at Harvard. So we were able to promote that. And because I think it's important to note that the synapses that we're preventing from elimination are still functional. And that was an important goal of our research project, to ensure that.
Thank you. So I think we're going to start wrapping up here because we're nearly at 1 p.m. on the East Coast. And we are getting quite a lot of questions about specific study site location, timing, eligibility criteria. And, unless you have specifics to share here, I'm just going to reiterate that a lot of those details are going to be available once there's more information about the trial, and HTSA will certainly share any information by posting it on HD Trial Finder, our clinical trials matching service, and I'm sure that more information will be available at clinicaltrials.gov. But if you have anything to add to that, since lots of people are wondering about whether they can participate, that would be great.
It's Enchi here, and I expect over the next couple months we will be giving more detail to HTSA and for your website as well as clinicaltrials.gov. So it won't be a long time. It's just in a couple months we'll be able to give more detail.
Excellent. Well, I want to thank you again so much, Enchi and Sanjay, for sharing all of this with the community and giving us so much time to answer questions here. Once again, I'll reiterate that that we're going to share this recording on our YouTube channel on HTSA's website, and it will be available again as part of our virtual convention on June 6th at 10 a.m. So thank you once again very much, and thanks everyone for tuning in today, and we'll see you next time.
Thank you.
Bye.
Bye-bye.
Bye-bye.